Hello students, I am Dr. Ashma Srivastav and today I will be explaining one experiment from the AKTU Chemistry Lab syllabus. The demonstration video of this experiment has also been prepared and it will be shared along with this complete explanation. Today's experiment is an acid-base titration that will be done with the help of an instrument called pH meter. The aim of the experiment is to determine the concentration of given acid solution by titrating it against standard alkali, that is NaOH solution, using pH meter. The acid that has been used or provided is acetic acid and its concentration has to be determined through this method. Now what we notice here in the aim is that in this titration we are not going to use any indicator. So it is an indicator-less titration between a strong base and a weak acid. The apparatus and reagents that are required for this experiment are as follows. Number 1, pH meter. Number 2, combined glass electrode, which is commonly called pH electrode. Number 3, buffer solution of 7 pH, which will be used to calibrate or standardize the instrument, that is pH meter. Number 4, the standard sodium hydroxide solution of known concentration, which we have taken as 0.1 m. And number 5 is the unknown acetic acid solution whose concentration has to be determined through this experiment. Now let us begin our discussion with the introduction. Acid-base titrations are commonly done with the help of acid-base indicators, but the accuracy and the precision of such methods is comparatively less than the titrations which are done with the help of certain devices or instrument such as pH meter and pH electrodes. When we do the titrations with the help of acid-base indicators such as phenolphthalein, methyl orange, then we have to stop the titration at one point where the desirable color change takes place, and that is called the end point. Now the perception of the desirable color change differs from person to person, and this results in error in noting down the end point value. Here in case of the titrations with the help of pH meter and pH electrodes, there is no such endpoint detection required because we will be noting down the values and then we will be plotting the graph to find out the completion of the reaction through the equivalence point. So here we are getting to know the more accurate point, that is equivalence point, and without detecting or judging any color change. So these titrations gives us more accurate and better results. Now this titration is an example of a potentiometric titration. Potentiometric titration is a volumetric method in which the potential between the two electrodes is measured as a function of added reagent volume to the analyte solution. There is no need of indicator, the titration is not stopped at the equivalence point, and the volume of the standard reagent consumed to reach the equivalence point is not seen through the burette, but it is determined graphically by plotting the titration curve. In this experiment, we have to determine the concentration of given acetic acid by titrating it with standard NaOH solution. Now the pH meter that we are using here is nothing but a electronic voltmeter which is capable of measuring the voltage of the galvanic cell in millivolts. The galvanic cell here is nothing but our combined glass electrode, which we will discuss in more detail on the next slide. Now the voltage of this galvanic cell is directly related with the pH, and this pH can convert that voltage into pH, which we are going to read out through the display. This pH value varies with temperature, so we have to set the temperature, room temperature, also on this pH meter. Now let us discuss in detail the combined glass electrode which we are going to use for the measurement of pH. This combined glass electrode is more commonly called pH electrode, and with the help of this electrode we can determine the pH of any solution in which is dipped. This combined glass electrode is a complete cell having two half cells. The first half cell is the glass electrode and the second half cell is the saturated calomel electrode. The glass electrode functions as the working electrode and the saturated calomel electrode is a reference electrode. The glass electrode is also functioning as anode, whereas the saturated calomel electrode, which is also abbreviated as SCE, functions as cathode. Now we will see this figure to understand where the glass electrode and saturated calomel electrode is present. Now this figure consists of two glass tubes. One is the inner glass tube and then the outer glass tube. Inner glass tube constitutes our glass electrode and the outer one is our saturated calomel reference electrode. This inner tube, which is our glass electrode, consists of Ag wire on which AgCl has been impregnated, and this electrode is dipped in the HCl solution of 0.1 m concentration. This glass electrode contains a special bulb at the bottom of it, and this special bulb is made up of special glass which is of high conductivity, and with the help of this special glass we are able to do this experiment or to detect the pH. The bulb of this glass electrode is made up of special glass which is going to detect the H plus ion present in its surrounding, and hence it helps us in detecting the pH of the solution in which is dipped. So the glass electrode is AgCl electrode dipped in 0.1 m HCl solution. This 0.1 m HCl solution is also present in the bulb of this electrode, and the bulb of this electrode or the glass of this electrode is made up of a special glass, and it is so fine that it is also called a thin membrane. So this thin membrane of the glass can read the H plus ion across the glass membrane, and that helps us in detecting the pH. Now let us come to the saturated calomel electrode. Saturated calomel electrode is present in the outer tube. It consists of Hg2Cl2 as a paste into which platinum wire is dipped, and the electrolyte is saturated KCl solution. Now here the cell notation for this complete combined glass electrode is given. AgCl dipped in HCl in contact with a special glass serves as our glass electrode, which is our working electrode. Working electrode means it is going to help us in detecting the pH. Then this glass is in contact with the test solution, that is our acid solution, acetic acid solution, and it will help us with the concentration of this acid solution has to be determined, and then there is a saturated calomel electrode which is also in contact with the test solution. The measurement of pH is based on the fact that when a thin glass membrane is in contact with the solution of different hydrogen ion concentration, here it is acetic acid, the potential difference develops across the thin glass membrane. Across this glass membrane there are two H plus ion solution. One is 0.1 m, that is fixed, which is present in the bulb, and the other one is on the outside of this bulb, that is present in the beaker, and that is our test solution. So potential difference develops across this glass membrane, and this magnitude of this difference, potential difference, depends upon the concentration of the hydrogen ions. Now the reference electrode which we are using here is having a constant potential and its value is 0.24 volts. So it is constant. So the potential of the cell depends entirely on the potential difference which is developed across this glass membrane and which is depending upon the hydrogen ion concentration outside the glass membrane, that is in the test solution. Now, once this voltage, the potential difference is read by the pH meter, it digitally converts that voltage into pH and we see the pH of the acid solution on the pH meter. Now, we will be recording the pH value of the titrating mixture from time to time by adding NaOH, and then we will be using those readings to plot a graph and to find out the equivalence point. What we are going to do? We are going to take a test solution. Into that test solution, our electrode will be dipped. Which electrode? Combined glass electrode. And then we are going to start the titration. We are going to add 0.5 ml of NaOH solution, and at every 0.5 ml addition, we have to stop and check the change in the pH values. And these pH values will be noted down. Now, what will be our observation? Our observation will be like this, that when we start adding NaOH to the acetic acid solution, then there is a increase in the pH, which is quite obvious. We are adding NaOH to acid, alkali to an acid, so the pH is definitely going to increase. But it follows one pattern, and what is that pattern? That initially there is a gradual rise, and then just near the equivalence point there is an abrupt rise in the pH value. After that equivalence point, then again when the acid is added, NaOH is added, the pH increases gradually. So, this is the pattern that is followed. Let me repeat it. When we start adding NaOH into the acid solution, then the pH starts increasing gradually. Then when we are about to reach to the equivalence point or in the range of the equivalence point, there is an abrupt increase in the pH, and after that abrupt increase, again there is a gradual rise. Now, here the mixture which we are using is acetic acid and NaOH. Now, the acetic acid and NaOH is a weak acid strong base mixture. So, the equivalence point of this mixture will be lying towards the higher towards the pH more than 7, and the jump of pH will take place in that region. So, let us now come to the procedure, and afterwards we will understand that what we are going to do with the readings that we have recorded. The procedure is followed in certain steps. Now, let us see them one by one. So, what we will do? We will set the temperature, room temperature, with the help of temperature knob on the pH meter, and then we will standardize this pH meter after setting the room temperature with the help of buffer solution of 7 pH. We will dip our electrode in the buffer solution of 7 pH, and then we will see what pH our instrument is showing for that 7 pH solution. If it is showing 7, then it is functioning at its best, but if it's showing the pH less than 7 or more than 7, then with the help of a knob which is called standardize or calibrate, we are going to set it to 7.00, and hence our calibration part is done. Secondly, what we will do? We will start our experiment now. So, step 2 is we will pipette out 10 ml of given acetic acid solution in a 100 ml beaker, and then we will be adding 40 ml of distilled water to this acetic acid solution, so that the most important part of our combined glass electrode, that is that pH sensitive bulb, is completely immersed in it. So, what we will do? We will take 10 ml of acetic acid solution, and then we will add distilled or deionized water, 40 ml distilled or deionized water into it to increase its volume, and into that solution we are going to dip our electrode. When we have added the distilled water, it has not affected the concentration of the 10 ml H plus ion. it is still the same. We have added water which is deionized, it is not providing any H plus or OH minus ion, and it is not going to affect the pH of our 10 ml acetic acid solution. That thing we have to keep in our mind. So, we have added 40 ml water, and now we will insert our combined glass electrode into the solution, and we have to keep in our mind that the bulb of that electrode is completely immersed in the solution. We will connect the electrode to the pH meter and we will record the initial pH of the acid solution. Then we will fill the burette with NaOH solution, the concentration of which is 0.1 m, and we will add 0.5 ml of NaOH to the acid solution, and then we will mix the solution and note down the change in the pH of the acid solution. We have to take down the readings at every 0.5 ml addition of NaOH, and then how long we have to take these readings? We have to take the readings up to that abrupt increase, and after that abrupt increase in the pH we have to take 4-5 more readings so that we can get a smooth graph which will be used to plot, which will be used to locate the equivalence point. We will be tabulating the readings and then we will be plotting the graph. The graph will be plotted between the pH change that is recorded and the volume of NaOH that is added. And from the graph we will determine the volume of NaOH that is utilized at the equivalence point for this titration mixture, which will be used for calculation afterwards. So, this is the observation table in which we have to record the readings. You can see that we are recording the readings at every 0.5 ml addition, and this process will continue until we observe one abrupt jump in between which our equivalence point should be lying, and after that we have to take 4-5 more readings. Now, when we are going to plot the graph, we are going to get this type of sigmoid curve. This titration curve has a characteristic sigmoid shape. The part of the curve that has maximum change in pH marks the equivalence point of the titration. So, this is the plot, or this is the graph which we are going to get when we are plotting the volume of NaOH added on the x-axis and pH change recorded on the y-axis. This is a graphical tangent method which is used to determine the equivalence point. Now, after plotting the graph, what we will do? We will plot two tangents. Now, the two tangents are the two parallel lines that are drawn to the vertical steep portion of the titration curve. The vertical steep portion of the titration curve you can see, and that is the region in between which we have to plot the two parallel lines which should be tangents. After plotting these two tangents at the upper and lower part of the sigmoid curve, we will be drawing one line perpendicular to both these tangents. Once this perpendicular is drawn, the midpoint of that perpendicular will be located, and from that midpoint one third line parallel to the both tangents will be drawn. The point where this middle parallel line cuts the titration curve, that is red in color, is our equivalence point, and the volume corresponding to that equivalence point we can see on the x-axis, and that volume is going to be used for doing the calculations or to find out the concentration of the acid. So, this is a graphical tangent method in which what we have done, we have plotted the graph between volume of NaOH added, which is on the x-axis, and pH on the y-axis. We have got a sigmoid curve. Then we have plotted two tangents. These two parallel lines should be tangents, and these two parallel lines are plotted in the vertical steep portion of the titration curve. After plotting these two tangents, we have to draw one perpendicular. That perpendicular has to be, once this perpendicular is drawn, then we have to locate its midpoint. After locating its midpoint, we have to draw one line parallel to these two tangents, and wherever this third parallel line is going to touch or cut our titration sigmoid graph, that will be our equivalence point. There is one more method for determining the equivalence point, and that is called the derivative method. And in that case, what we do, the x-axis is having the same thing, that is volume of NaOH added, but the y-axis is having the derivative of the pH, which means that we are going to plot the change in pH by change in volume, and then we are going to get a blue type of graph. The tip of that graph is going to give us the equivalence point, and we can see its volume corresponding to that tip on the y-axis and the pH on the x-axis. Sorry, pH on the y-axis. Now let us see this graph again. The volume of NaOH consumed at the end, at the equivalence point, we are going to get from the x-axis, and the pH of the mixture at the equivalence point we will get to see from the y-axis. And since it is a weak acid and strong base mixture, so the pH equivalence point pH will be more than 7, and you can see on the figure that it is shown as 8.72. So 8.72 is the equivalence point pH for this mixture. Now let us come to the calculation. The calculation has to be done by using that same molarity equation, that is M1V1 is equal to M2V2. M1V1 is for acid and M2V2 is for the base. Now what we have to determine? We have to determine M1, and rest of the things are known to us, which means that M2, that is the molarity of base, is 0.1, which we know, and the V2 is the volume of NaOH. Now from where we will get the volume of NaOH? We will get the volume of NaOH at equivalence point from that graph, and that extrapolation of the equivalence point to the x-axis is going to give us the V2 value. So we will take that V2 value from the graph, and then it will be substituted here in the graph in this formula to get the molarity of the acid solution. And afterwards, at the end, we will report the result in this way. The first thing that we have to report is the pH of the given acetic acid solution, and that is the pH of acetic acid solution before starting the titration, before adding NaOH to it. Second thing is, that is the main part of our experiment, that was the aim of our experiment, that is concentration of the acid. So this concentration we are going to get from the calculations which we have done just before on the previous slide. And after that we have to report the pH at equivalence point. This also we are going to get from the graph, and that we are going to put it over here. One thing that we will keep in mind, since it is a weak acid strong base mixture, so it should be above 7. If it is coming below 7, then there must be some error in plotting the tangents and locating the endpoint. And lastly, we will be reporting the room temperature, which we have seen with the help of thermometer that has been provided to you. And that finishes the complete explanation of this experiment. Thank you.